Okay. Hello, everybody. My presentation is going to be on animal drug and psychological testing. To begin, I'd like to ask the audience, who here takes Advil, Tylenol, or any form of allergy medication on the regular? Because personally, I know that I do. Was everybody aware of the fact that before these medications hit the counter, they underwent years and years of animal drug testing to ensure the safety for humans? But what is animal drug testing? This form of testing is the use and experiment of non-human organisms for the purpose of better understanding the human mind and body. This first began with Greek physicians such as Aristotle and Erasistratus in 300 BC for the purpose of understanding human anatomy, physiology, microbiology, and pharmacology. This really became important in 1938 with the release of an antibiotic called elixir sulfanilamide, which led to mass poisoning and the death of over 100 people across 15 different states. As you can see here, the states primarily resided in the southeast region of the United States, but did spread all the way over to California. Because of this, the 1938 Food, Drug, and Cosmetic Act was released to prohibit the production of medication without prior animal testing, according to National Institute of Health. Since then, animal testing has only grown more significant, with over 115 million animals tested on per year across over 1,200 different animal testing sites, according to Humane Society International. In this PowerPoint, I'll be covering three main points: animal drug testing for over-the-counter and prescription medications, animal psychological testing, and the purpose behind all of it. Beginning with my first main point of animal drug testing. But what is animal drug testing? This form of testing is a stage in a clinical trial where over-the-counter and prescription medications are forcefully ingested by animals and carefully studied to monitor the effects before moving on to human trials. In this type of study, two species, typically dogs and rats, are given high doses of medication for 30 to 90 days and carefully monitored for organ drug toxicity. These tests can last up to 4 years to test for long-term disorders such as infertility or cancer treatments. These tests are also heavily mandated by FDA laws. Common medications include penicillin, cancer treatments, insulin, vaccines, and much, much more. The environments of which these tests take place range significantly. Most facilities have proven to keep their animals in unhygienic cages, instilling fear and sadness on these creatures, while other facilities, such as Pfizer facilities, have promised to keep their animals in a clean and open environment, as well as providing a healthy and hygienic diet. Possible tests that may be included are chemical exposure, forceful injection, gas inhalation, and genetic modification. In fact, over 90% of these drugs in clinical trials actually fail due to animal testing revealing unsafe and hazardous environments for humans. This is according to science.org. The majority of these animals in these tests are either killed, but the ones that do survive are still killed after to test for organ toxicity and inspection. Additionally, the ones that do survive are typically used for multiple experiments following that initial one. Moving on to my second main point of psychological testing. What is this type of testing? In this sort of testing, animals undergo severe mental and emotional stress to further understand the human mind. Animals have greatly assisted in developmental contributions for therapeutic and psychological advances in medication. Our knowledge of thirst, hunger, sexual desire, fear, sadness, and joy was achieved with great help through animal testing. Common experiments include electrical shocks, anxiety-inducing medication, food deprivation, mother separation, and brain function altercation to affect their behavioral, sensory, and cognitive capabilities. I want to provide an example of this form of testing. In 1967, two University of Pennsylvania students developed a facility with two enclosures separated by a small but accessible hurdle. On one side of the exposure, here on the left side, there were electric charges on the floor, but not on the other side. These students discovered that when dogs receive electrical shocks that they cannot control, they're clearly presented with severe stress and anxiety, but not when they learn that they can control the situation by jumping over to the other side. They discovered that when dogs learned this capability, many of them didn't even try jumping on the other side, as their stress and anxiety was significantly reduced and they did not make it a priority for them to get to the safe side. A similar experiment was done with rats. Once the rats learned that they can get over to the safe side, many of them wouldn't even try moving over to the safe side, and they would stay on the electrical shock side. This significantly helped with our understanding of serotonin and how once these dogs learned they were in control, serotonin flooded their brain, relieving their stress and anxiety. Dogs have shown great recovery when they're put up for adoption. As I stated previously, the majority of dogs are either killed afterwards, or used for multiple experiments, but the ones that are put up for adoption have shown great success in recovery. According to Oregon Somatic Therapy, these animals are free of PTSD-like symptoms. Without this ability in the wild, they would simply shut down, be uptight, confused, and they would fail. They simply wouldn't survive in the wild. Animals tend to release their trauma. Moving on to my third point, the purpose behind all of this. Why do we do it? Without animal testing, our state of medical advancement may not have been anywhere near where it's at today. It's nearly impossible to verify the safety of these medications without testing them on live subjects first. It's simply too risky to test them on humans, let alone illegal. As we stated previously, the majority of clinical trials actually fail. As you can see in this graph, only 6 out of 100 possible drugs actually became drugs for the public. The rest of them failed due to either being ineffective, not reaching the target, having toxicity levels that couldn't be managed, or failing preclinical tests. This allows for scientists to control factors that may affect the outcomes of experiments and allows for a more precise understanding of psychological factors involved. Additionally, it provides a greater certainty when developing medications for the actual treatments themselves. Not only does this help develop medications, but is essential to the discovery and causes of these actual diseases themselves. Stanford Medicine summarized this very, very well by saying that the use of animals in some forms of biomedical research remains essential to the discovery of causes, diseases, and treatment of diseases and suffering in humans and in animals as well. While this is all very important, there are a couple of alternatives that still need to be highlighted. We've got the use of isolated cell tissue that is not in a live organism, using computers and math to simulate these experiments, finding ways to make these experiments safe for humans, which is unlikely for now, and using bacteria instead, which actually is not as effective as using animals. Animal testing is an incredibly controversial subject. While it's important for the medication development and therapeutic relief, the act of putting animals in harm's way strikes a lot of controversy for better alternatives. The next time you're using your regular medications, ask yourself, is this medication and its ensured safety worth the suffering of millions, or is it considered a grand act of severe animal cruelty that needs to stop immediately? Thank you.